During mitosis, chromosomes are segregated by a microtubule-based structure called the mitotic spindle. However, as Helder Maiato from the University of Porto in Portugal explains, the spindle is embedded in a matrix of proteins that help it carry out its function.
Many proteins have been found to accumulate in this area in a microtubule-independent manner. So in this study, we set out to investigate what drives the accumulation of these proteins in the spindle region in a microtubule-independent manner.
In Drosophila cells, for example, the nuclear pore protein Maiato, which regulates the spindle assembly checkpoint, is retained around the spindle in early mitosis, even when spindle microtubules are depolymerized. And after nuclear envelope breakdown, soluble tubulin diffuses into the spindle region, even if spindle assembly is inhibited. Maiato and colleagues, led by graduate student Nina Schweizer, thought that membranes derived from the nuclear envelope and endoplasmic reticulum might help proteins like Maiato and tubulin accumulate around the spindle in a microtubule-independent manner.
So it was known for many years that the spindle region is surrounded by membranes. Many of these membranes derive from the endoplasmic reticulum, but the function of these membranes have been quite mysterious. So we looked at membranes with the best tools that we have available today in live cells. And indeed, the spindle region of both Drosophila and human cells are surrounded by membranes, which we use the classic term spindle envelope.
Disrupting the spindle envelope by laser microsurgery impaired the retention of Maiato and the accumulation of soluble tubulin in the spindle region.
These membranes do a very important task, which is to exclude from the spindle region all the large organelles in the cytoplasm, such as mitochondria.
There are, however, gaps in the spindle envelope that would allow the diffusion of small molecules such as soluble tubulin and the spindle checkpoint protein MAD2. Mathematically modelling the diffusion of these proteins revealed that they would preferentially accumulate around the spindle because this region is less crowded with large organelles.
So because it's less filled with organelles, you have more volume to be occupied. The molecules can accumulate into higher levels in the spindle region compared to the cytoplasm, in which these large organelles fill a lot of space.
Thus, by excluding large organelles, but allowing the diffusion of mitotic regulators, the spindle envelope helps these proteins to accumulate around the mitotic spindle. At the same time, the spindle envelope helps to retain larger protein complexes, such as the one containing Maiato, in the spindle region. To test whether this is important for the assembly and function of the mitotic spindle, Schweizer et al. Again used laser microsurgery to disrupt the spindle envelope in early mitosis.
And the first thing that we noticed that when we make these holes, this creates a local area of accumulation of tubulin, for example, that allows ectopic formation of microtubules that will contribute to the formation of ectopic spindle poles and will give rise to multipolar spindles. And this is quite a deleterious condition because multipolar spindles will not be able to segregate chromosomes in an equal way. And this is exactly what we find. We see a significant chromosome missegregation once we do not confine spindle assembly within this spindle envelope.
Thus, by facilitating the accumulation of mitotic regulators in a matrix around the spindle envelope helps the spindle to assemble and segregate chromosomes correctly. It remains to be seen exactly how some of these spindle matrix proteins, such as Maiato, promote the fidelity of chromosome segregation, but, for now, you can learn more about how an organelle exclusion envelope assists mitosis and underlies distinct molecular crowding in the spindle region in the paper by Schweizer et al., published in the August 31st, 2015 issue of the Journal of Cell Biology.